So my name is Tune Pers. I'm a group leader here at the center, and it's a great pleasure to introduce today's speaker, first speaker, Professor Randy Seeley from the University of Michigan. So Randy's work really focuses on understanding the action of peripheral hormones on energy homeostasis and circulating fuels. And Randy is running a major effort and has been publishing a number of papers also on elucidating and finding out the role of figuring out why gastric bypass has a number of beneficial effects on metabolic health. And Randy has made several contributions to science. Among others, he has published studies helping to understand the field while GLP-1 receptor agonists are having a very beneficial effect on weight loss. And he has shown that this is mediated through the CNS, and he has shown mechanisms through which this happens. And also, as Randy has shown, that bile acids are very important mediators of bariatric surgery on metabolic health. So it's a pleasure to have you. I just wanted to say, those of you who have been in a major bar in Michigan will know that there's even a drink named after Randy. It's called Lemon Seeley. We won't have any of those today, but I'm sure we'll have great fun listening to a talk and interacting with you afterwards. Randy, it's a pleasure to have you, and the floor is yours.
Thank you very much. All I can tell you is that lemon Seeleys are an acquired taste, and dose is very important. Right? So thank you so much for the invitation to come. This is my first trip outside the United States, so I'm super happy to be here in so many ways. I will start by just pointing out again my Twitter handle, right? For those of you who want to engage me on Twitter, I'm happy to do so. So if anything interests you, let me know. I also start by pointing out my disclosures, right? We work with a lot of different companies that are trying to bring therapies to patients, and particularly with the work you're going to see today, right? Large hunks of it were supported by Novo Nordisk. Okay, so bariatric surgery, right? So these are the two most common bariatric surgeries. One is called vertical sleeve gastrectomy. The other is a Roux-en-Y gastric bypass. And I got interested in these because the benefits to patients are so large, right? These are by far the most effective therapies for either obesity or type 2 diabetes, though the drugs are getting better, right? We're getting closer and closer. And I would talk to surgeons about why this happens. And the explanation they would give me made no sense to me, right? So the way they would talk about this, right, is the idea that what happens after these procedures has to do with a mechanical change in how they work. So there we go. Okay. So this is vertical sleeve gastrectomy, where we've removed 80% of the stomach along the greater curvature. This is a Roux-en-Y gastric bypass, where we've made this small pouch right at the top, and then the food is now deflected from going through its normal path through the small intestine and is diverted into the jejunum. So this procedure is often referred to as a restrictive procedure, right? The idea that we make your stomach so small that you won't or can't fit more calories down your gullet. And this procedure is referred to as restrictive, right, because you have this small pouch in theory, and malabsorptive, right, because we're bypassing part of the intestinal components, right, that would normally absorb calories. Again, the idea that malabsorption is important, right, is the idea that we somehow sneak calories out of your rectum without you noticing, right? That is not in the brochure, right, for surgery. But again, that idea made no sense to me, right? So if I snuck calories out of you, if I made you malabsorptive, would you be more hungry or less hungry? Most people would be more hungry, right? If I made it impossible for you to eat because I wired your jaw shut, right? I made it restrictive. Would you be more hungry or less hungry, right? And the answer is you'd be more hungry. But what happens after surgery that makes it so different than everything else is that patients lose a lot of weight and they're less hungry, right? So that implies, right, that these mechanical explanations for what it is that surgery does just don't make any sense, right? There has to be something else. So much of what my lab does is to search for something else. The way we've done that, right, is to build rodent models of these particular procedures that allow us to explore their mechanism of action. And I'll just point out, again, these rodent models for surgery are really important, right? And they're more important than when you think about pharmacology and pharmacological interventions. Why? Because you cannot do a double-blinded randomized control trial for surgery. Nobody wants their surgeon to not know what surgery they're doing while they're doing it, right? So the point is that what you see in the clinical trials, right, is a product of not only what the biology, right, but what the patient expectations are. They know they had surgery, right? The rodents don't know what's supposed to happen, right? They get no instructions after the procedure. I do not have a rat registered dietitian telling them what to eat, right? Everything you see is a product of the biological effects of changing their GI tract. So this happens to be a rat version. I'm going to show you a bunch of mouse versions as we go forward. And what you see here is animals who have either been maintained on a high-fat diet or standard chow, right? And that middle bar are animals, this is before the surgery, are going to get surgery, right? And 35 days later, when you come back and measure their body fat, the animals have lost 50% of their body fat. They've lost all the excess body fat that accrued as a function of being on that high-fat diet, even though they're still on the high-fat diet, right? How do they get there? How do they lose this weight? Well, the answer in case of vertical sleeve gastrectomy is largely because they reduce their food intake. So this is what you see, right, in the aftermath of the surgery. What you can see very obviously, right, is that in the immediate aftermath of surgery, the animals eat less. Great. But what you can also see is that these animals go back to eating what they did before. Where's the restriction, right? And I can tell you that this has nothing to do with their stomachs dilating, right? Some physical change that allows it. Their stomachs look exactly the same on day 17 as they did on day 8. So again, what's happening here? How do we try to explain this? And again, we've spent our time trying to understand what the molecular mechanisms that might be. And again, when you look at these procedures, what's interesting, right, is that the two procedures look very different, right, from a sort of a surgical technique point of view, right? You're doing very different things to the intestine, but you end up with clinical phenomena that look very similar, right? Large reduction in body weight, large improvements in glucose regulation. So for us, what we've been trying to do is to look for commonalities between the two procedures that would help us explain what it is that happens. And when you do that, you get a whole bunch of things that are possible, right? And one of those things is existing gut hormones that change after surgery. So GLP-1 and ghrelin are examples of this that have been hypothesized to be important components of what happens. I can tell you that in our hands, when we do loss of function genetics and do surgery on mice that have loss of function of those proteins, it does not matter. The animals respond normally. Those are not necessary to the benefits of surgery. On the other side of this slide, right, are a variety of things that we've already published that are necessary for the surgery in different ways. And again, I won't bore you with that because that's all published, right? What I want to talk to you mostly about today is some unpublished work that focuses on another protein called reg3 gamma. And I'll give you a little explanation about what that is in a second. So one of the things you have to understand about the GI tract, right, is that it has this complex a set of functions that are not compatible with each other in the normal way, right? So your GI tract has two big functions, right? One is to keep bad things out, right? To be a barrier, right? So like your skin, right, that barrier is turning over all the time, right? So again, it's turning over every 7 days. But the second thing it has to do is it has to absorb things, right? You got to have nutrients, micronutrients, water, all the things that you have to bring in from the outside have to come in through your GI tract. And again, the balance between being a barrier and being absorptive, right, is a constantly evolving and changing thing for the GI tract. And again, for omnivores like humans, right, that change what they eat every day, right, it requires a number of potential changes and balancing between those two activities. We refer to that as enteroplasticity, the idea that the GI tract can change, and it's a very plastic organ compared to most of the organs that we have in the body. Now, again, so what I'm going to tell you about, right, is the idea that these surgeries change GI functions, right, that relate to these components of what the GI tract has to do. And I want to tell you about the role of various proteins in that. Right? Again, and the first thing that happens after these surgeries that's really obvious is you have this big increase in gastric emptying rate. So these are essentially rat barium swallows And what you see is what happened 5 minutes after we gave them a gavage of Ensure with barium in it. And what you see in the sham animal, right, is that almost everything is still in the stomach, right, 5 minutes later. If you look at both vertical sleeve gastrectomy and root-and-wide gastric bypass, 95% of what we gave the animal is already past the stomach and into the intestine after the surgery, right? Again, where's the restriction again, right? There is no restriction, right? Because everything is actually flowing really rapidly. It's not like your stomach is filling up and stretching, right? Things are flowing back out into the intestine. So how do you think about that? Now, again, that has to cause changes downstream, right? You no longer have a stomach that's protecting your intestine and meting out calories in a slow, easy pace. Now your intestine has to face this incredible barrage, right, of intestinal, of chyme, right, that's coming at it at a very fast pace. And that requires, again, this enteroplasticity, right? And one of the things that has to change is the barrier function of the GI tract. And again, when we think about the barrier, we probably think mostly about the physical components of the barrier, right? You get tight junctions between cells that make it hard for things to wiggle out. You've got mucus production, right, that helps plug gaps, right, to make it hard for bacteria or other bad things to wiggle out. But there are absolutely chemical components of that barrier that are absolutely necessary, right? And among these are antimicrobial peptides, peptides that are secreted by Panna cells along with the mucus that actually are really important for segregating your gut bacteria from the host. One of those is reg3 gamma, right? And a family of these antimicrobial proteins that are all reg proteins. So when you look after bariatric surgery, right, and you use RNA-seq to just look at what are the transcriptional changes, right, that are common between VSG and root-and-wide gastric bypass, what you see is the top item that comes out are these antimicrobial peptides when you look in the small intestine. Again, if you look in a very specific way, right, look at all of these, right, again, reg3 gamma, right, becomes clearly upregulated as a function of these surgeries. So that's how we started in this, right? I didn't know anything about antimicrobial peptides or regs when we started this project. But this is compelling to believe that, in fact, that maybe they have an important function. For those of you who don't know, these reg proteins, again, are expressed in lots of different tissues. And their name and where they were identified is primarily in beta cells, right, in pancreatic resection models, right, where you have growth of beta cells, right. These are highly upregulated, and reg3 gamma is one of the most upregulated in the pancreas in those kinds of situations, right. So it's definitely involved, again, in plasticity, right, in change that has to happen in lots of different tissues. So what you see first, right, again, when we look at diet-induced obese animals, right, you get a profound downregulation of reg3 gamma, what you see here. If you look in OB mice, right, again, they're obese because of a lack of leptin, same thing, right, a dramatic decrease in the amount of reg3 gamma that's expressed in the GI tract. Now, again, if you look at a root-and-wide gastric bypass, you can see this upregulation of reg3 gamma. If you look at vertical sleeve gastrectomy, you see the same thing, right? So again, we know that this is being upregulated. And now the question is this a hormone, right? Does it actually circulate at levels that might matter? I can tell you that's actually been a very hard question to answer because the assays stink, right? And we went through a lot of assays to try to find one that actually works. I won't bore you with the details of it, just know that it's not fun. But when you use this not fun assay, what you see is a clear increase in circulating levels of reg3 gamma, right? So again, it's possible then that means that reg3 gamma could have a role inside the lumen, right, as part of the way it's an antimicrobial peptide, but also that in fact it's a circulating hormone that's secreted from the gut and can act on distal portions of the metabolic structures. If you look in humans after vertical sleeve gastrectomy, you can see there's a wide variance, right, in the circulating levels of reg3 gamma. This happened to be in adolescents, but you can see a very clear increase in reg3 gamma. Again, in the analog of reg3 gamma, the human analog of reg3 gamma, that's upregulated after vertical sleeve gastrectomy. Okay, here's the guts of it, right? So we now know it's changing, right? Does it matter, right? And the way we have done this, right, is to use loss of function genetics in these animals and ask the question, right? If reg3 gamma is really important for how surgery works, then in animals that lack reg3 gamma, surgery should be less effective to cause improvements in glucose and weight and all the other things, right? So again, this is an experiment done by Jae-Hoon Shin in the lab, again, who's led almost all of this stuff. And This is before surgery. Again, I'm just normalizing things to make it a little bit easier, right? Again, everybody's at 100%. And what you see after surgery, right, is that in the wild-type animals, in this case, they've lost about 25% of their body fat. And you can see in the animals that lack reg-3 gamma, that effect is entirely absent, right? So there's something about reg-3 gamma that is necessary for the benefit to weight that happens after vertical sleeve gastrectomy. If you look at fasting glucose levels, right? So now let's look at the glucose side of the equation, right? You can see a decrease in fasting glucose levels that happens after vertical sleeve gastrectomy. Again, entirely absent in the animals who lack reg-3 gamma. And again, point out here, right? The surgeons made the stomachs just as small in the mice that had lacked reg-3 gamma as the wild types, right? Having a small stomach is not sufficient to drive these benefits of vertical sleeve gastrectomy. Right? In the absence of reg-3 gamma, it doesn't matter how small we made the stomach. Right? Here's glucose tolerance. Right? So again, looking at glucose tolerance and reg-3 gamma knockouts, and you can see a nice effect of vertical sleeve gastrectomy to improve glucose tolerance. Again, that is much reduced in the animals who lack reg-3 gamma. This is easier to see when you look at the areas under the curve. You can see, again, nice effect of vertical sleeve gastrectomy that's absent in reg-3 gamma knockouts. One of the important things that bariatric surgery does, right, is to improve insulin secretion, particularly postprandially, right? So again, you get much bigger responses to glucose loads in terms of insulin secretion. So these are baseline insulin levels. You can see at 15 minutes after a gavage of a mixed meal, you can see this nice improvement in insulin secretion, right? So you get much better postprandial insulin secretion. Again, entirely absent in the animals that lack reg-3 gamma. One of the interesting things about this phenomena is that it survives when you take these islets out of the animal, right? So if you take these islets out of the animal, put them in a dish, what you see is that you still get those islets have an increased response to glucose. But again, islets from animals that lacked reg-3 gamma do not have that phenomenon. So again, reg-3 gamma is necessary to the benefits of vertical sleeve gastrectomy to alter what happens at the level of the islet. So again, bariatric surgery has a bunch of other effects, again, that at least some of them are probably weight independent, and that includes benefits on the liver. So here what you see is that bariatric surgery produces this nice decrease in liver fat in DIO animals. Again, that's entirely absent in the reg-3 gamma. So that's all about the metabolic effects, right? That reg-3 gamma is really important for these metabolic benefits of the surgery. But the question becomes of the changes in gut function, right? How many of these things are also dependent on reg-3 gamma? So again, we're going to show you assessments of gut barrier function and the changes that happen after surgery, right? And one of the things that surprised us, right, is that what you see after surgery is an actual improvement in the barrier, right? And the way we measure that, right, is we give the animals dextran loaded with FITC. And FITC, again, is something again that you can just measure in the plasma, right? And the amount in the plasma reflects how well the barrier worked, right? Lower amounts of FITC, better barrier, right? And what you see after vertical sleeve gastrectomy in normal mice, right, is you get this really strong improvement in the barrier. And that makes some sense, right? Because you no longer have the stomach being a barrier and providing protection. That you actually have to tighten up your function of the GI tract in order to maintain some level of barrier in terms of keeping out bacteria and other problems, right? But again, that's entirely absent in the animals who lack reg-3 gamma, right? So again, this component of the barrier, right, is critically important to why it is that surgery can improve the barrier. And that shows, again, that this is not just about its antimicrobial nature, right? It's about that reg-3 gamma is orchestrating other things about what the GI tract does, again, and in response to various aspects of what you eat, right, and what the biome is doing, right, what the bacteria in your GI tract are doing. So this extends to various aspects of cellular stress responses that are regulated by reg-3 gamma. So this is an intestinal organoid, right, where we're now stressing them out by giving them, by throwing them in hydrogen peroxide. And again, you can measure reactive oxygen species, all sorts of other sort of cellular stress responses. And again, you throw hydrogen peroxide on them, right, it goes up. And what you see is that in the absence of reg-3 gamma, that effect of hydrogen peroxide is worse, right? So again, reg-3 gamma is part of a component that actually limits some of the reactive oxygen species. And that if we add back reg-3 gamma into animals, into organoids, right, that lack reg-3 gamma, you can actually ameliorate that deficiency, right? So again, intestinal organoids, right, all by themselves, right, respond in terms of their cellular stress response, they respond to reg-3 gamma. And again, does this extend to the in vivo situation? And it turns out, right, that when you look at animals and take out their intestine and look for various markers of cellular stress, you can see that vertical sleeve gastrectomy is actually lowering it and does so again in a reg-3 gamma dependent fashion, right? That effect is absent in reg-3 gamma knockouts. All right. So now I've told you all this stuff about vertical sleeve gastrectomy and reg-3 gamma. It makes no sense, right, that reg-3 gamma is here to mediate vertical sleeve gastrectomy, right? That there's no evolutionary pressure to respond to what a bariatric surgeon and how they want to make their money, right? So the question is, what is this system actually for, and why is it responding in this way? So we began a search for looking for other things that we knew would improve GI function, right, and are associated with improved metabolic function. And the question is, do they have in common the ability to regulate reg-3 gamma? So what I'm going to show you are animals that are put on a high inulin diet. So this is pulled from the Cleveland Clinic, just because I think it's funny. And again, it is a soluble fiber that it says it can prevent overeating. We'll get into that in a minute. But these soluble fibers change the microbiome dramatically and can be used to improve GI function in a variety of situations, right? So again, we're going to take animals, put them on a high inulin diet, and see what happens. And the first thing you see, right, is you get a dramatic upregulation of reg-3 gamma, right? And that extends actually to the circulating levels as well. And again, when you use these high fiber diets, you can ameliorate some effects of high fat diets, right, to cause metabolic disruption, right? So what you see here, right, is the ability of inulin diets, right, to improve glucose regulation in animals that are maintained, again, on a background of relatively high fat, okay? Now, again, what you see as well, right, is that effect is greatly reduced in mice that lack reg-3 gamma, right? So again, this is another reg-3 gamma dependent function. So here's, again, a role of changing the GI tract by changing the diet, right, that provokes reg-3 gamma, right? And again, in reg-3 gamma knockout animals, the response is less. Now, what about, again, GI function? Right? So again, is there a relationship between GI function and reg-3 gamma after these diets? So again, this is Fitzy-Dextran. This is showing you, again, the improvements, the relative improvements you see on the inulin diet. And you can see that in the wild-type animals, they improve, and the improvement is less in the animals who lack reg-3 gamma. And then the other graph here, right, is to show you that the tight junction proteins that are associated with improved barrier function, again, go up after the soluble diet. And again, that effect, right, is dependent on reg-3 gamma, right? So again, it's not just reg-3 gamma as a barrier, as an antimicrobial peptide, right? Again, it's regulating different aspects of the barrier as well. Okay. So again, I've said it's an antimicrobial peptide. So what's the role that the microbiome plays in the roles of, again, after vertical sleeve gastrectomy, these high inulin diets and such, right? And where does reg-3 gamma fit into this? So the first thing I have to say to you, right, is the majority of the work on the microbiome focuses on the colon. And there's good reasons for that, right? One is where most of the bacteria are, right? As a matter of, there's orders of magnitude larger numbers of bacteria that hang out in your colon than in your small intestine. It produces most of the metabolites because it's got the bulk of the bacteria. Then you'd expect, right, that most of the metabolites that are derived from the bacteria that we think of as part of how the host responds, right, come from the colon. There's also a lot of looking under the lamppost here, right, because it is way easier to sample, right, the bacteria in the colon than it is in the small intestine, right? Again, particularly when you're talking about human studies, it's really easy to get fecal samples, right? It's very hard to get samples from the small intestine. But that doesn't mean, again, that the reason that the microbiome might be important is really centered on the colon, right? After all, again, much of the business of what's happening in the intestine is really happening in the small intestine. And certainly its relationship to the larger metabolic control, right, you would think of has to do with the small intestine rather than the large intestine. Again, so if you look in the small intestine, you get much smaller population of bugs. They're more diverse. their close proximity to the adaptive gut functions that we're talking about here, right? So again, those bacteria are in close contact, right, to these different aspects of gut function that we might think are important, but they're much harder to sample, right? Again, so I'm going to focus you on the small intestine here, right? And when you do that, and you look at what happens to the bugs after vertical sleeve gastrectomy, one thing you see is a big increase in lactobacillus. And again, there's two parts of this, right? So again, one is that there's this big increase in lactobacillus under these different circumstances. But what you also see is that in reg3 gamma knockouts, the small intestinal bacteria look the same, right? It is not the case, right, that by getting rid of reg3 gamma, we've changed the nature of the bacteria that are hanging around in the small intestine, which is in some ways counterintuitive, right? I introduced this as an antimicrobial peptide. Right? You'd think that, in fact, it would have an influence. But the reality is, again, it's one of many antimicrobial peptides. And so the bacteria are not likely the target of reg3 gamma, right? So the issue becomes now, is reg3 gamma downstream of the bacteria? Again, if you look after vertical sleeve gastrectomy, back up here, right? So again, vertical sleeve gastrectomy results in this big increase in lactobacillus. Lactobacillus, right, are probiotics, right? I guarantee you that there is a store here, right, that you can go and find and you can buy probiotics with lactobacillus. Again, these probiotics are used in a variety of circumstances to improve metabolic function in some cases, but also to improve GI function, right? That's what they're often prescribed for. So what did we do, right? We took probiotics, right, that included lactobacillus. This is called VSL number 3. The L stands for lactobacillus, right? It has several different flavors of lactobacillus in it. And what you see is when you start giving animals daily gavages of this probiotic, what you get is an increase in the plasma levels of reg3 gamma and an increase in the intestinal expression of reg3 gamma. Again, does that matter to anything that happens? Again, so this is intestinal permeability. Again, it's clear, right, that I won't bore you with the details of the experiment because we do a bunch of things to make the animal's barrier function worse, right? We challenge them in different ways. And what you see consistently, right, is that VSL number 3 will reduce the ability for these bad things that we've done to the GI tract to reduce barrier function. But again, is that dependent on reg3 gamma? And the answer is absolutely, right? So again, reg3 gamma is important for vertical sleeve gastrectomy. It's important for high soluble fiber diets. It's important for this probiotic, right, to exert these effects. Again, so this is, again, is a different version of this probiotic, right? But again, what you see is that you get an increase in reg3 gamma levels. And again, in this other version of it, right, you can also see the fact that we get an improvement in the gut barrier, right? That again now is dependent on reg3 gamma. Right? Okay. All right. All this is super fun. The question becomes, how do you use this, right? And again, there's a really important question to be answered here, right? So, is the important function of reg3 gamma to act inside the lumen, right, to regulate various aspects of gut function, including the barrier? Right? Or is it, again, we know that it circulates, we know that circulating levels change, right? Or does it act as a circulating factor? And one way to try to test that, right, is to give reg3 gamma in the periphery, right, and see what happens, right? Now, again, I will tell you that this is where many scientific endeavors go off the rails, right? Because making these proteins are not trivial. Going to a catalog and ordering them does not guarantee you that what's in the vial is actually what you hope for. Right? So in fact, this is, again, where part of the work with Novo Nordisk has been certainly important. Again, I'll recognize Sally Lyons-Abbott, right, who's a protein engineer who helped us make various forms of reg3 gamma. And again, what you see here, right, is that in a normal animal maintained on chow, you give them a glucose tolerance test, right, and add on pharmacological doses of reg3 gamma, you can see this improvement in glucose regulation. Again, it's consistent across different preparations, right? So again, it's a small but reliable effect that we see. Now the question again, so is that effect because it acts in a way on distal organs associated with glucose regulation, right? Or is it acting on the gut in some way, right? So again, how do you answer that question? Well, so as I said, reg3 gamma is an antimicrobial peptide. And how does it do that? Well, it essentially, it punches holes into bacteria, right, to kill them. That's not a receptor-mediated phenomenon, right? The question is, are there receptors for reg-3 gamma that matter? There have been hypothesized receptors, including this one called EXTL3, okay? And again, in a variety of cellular assays, right, where you look at different functions of reg-3 gamma, you can see that EXTL3's activity is important for mediating the effects of reg-3 gamma. So what do? Right? We took mice that were, where we knocked out EXTL3 out of beta cells, right? And now repeat the pharmacological experiment with, by giving reg-3 gamma, right? And what you see, again, is that in normal animals, right, this administration of reg-3 gamma causes a nice improvement in glucose tolerance. But what you can see is in the mice that lack EXTL3 in the pancreas, you lose that effect. So the implication, right, is that at least for some of the benefits of reg-3 gamma when delivered pharmacologically, right, it requires EXTL3 downstream in the pancreas. And I bore you with that because it implies, right, that there is potentially two different ways in which reg-3 gamma might matter, right? There's the idea that it's acting in the lumen, but that, again, this at least opens up the possibility, right, that it acts as a gut hormone as well. So what I've tried to convince you today, right, is that reg-3 gamma sits in the middle between the organism and its bacteria, right? That alterations in various things, right, that will change the gut microbiome, right, are read out as changes in what happens in reg-3 gamma, right? And that change in reg-3 gamma can do two things, right? One is it can improve metabolic function. The other is that it can improve gut function. Right? Now, what I think of as the $100 million question, right, then becomes, what's the relationship between the gut function and the metabolic function, right? Is it sufficient to think, so again, dual actions, but the question is, what's the relationship between these things that are happening in the GI tract and what's happening in terms of metabolic regulation? What is the degree to which the diets that we consume that we know deleteriously impact metabolic regulation depend on the ability to alter and, again, compromise the function of the GI tract? So that's what we now spend a lot of our time trying to understand. And again, the question becomes whether if bariatric surgery improves gut function under these circumstances, right? Again, how much of what bariatric surgery does is a result of that improvement in gut function, and can it be targeted, right? And again, that's where we spend most of our time now. So I will stop there and point out the obvious, which is I didn't do any of the work you just saw, right? Again, much of this work was led by Jae-Hoon Shin in the lab, right, who's done a fantastic job of pushing this project over the last 4 or 5 years, I'm embarrassed to say. And again, I mentioned Sally Lyons-Abbott at Novo, again, but everybody in the lab plays an important role in this. Again, I just want to thank everybody for your attention and how nice it is to be anywhere, but certainly particularly to be here. So thank you very much.
Thank you very much, Randy. So we'll have time for a couple of questions from the audience, and we'll start over here and then go through here.
Andrew Paulson, Copenhagen. I was fascinated by the effect of protection on the beta cell when you take out the islets. And I basically have 2 questions. One is, what do you think is the mechanism? Is it metabolic imprinting? And have you done the similar experiment in your reg-3 gamma treated mice to see if that effect is preserved ex vivo?
Yeah, that's a really good experiment that we haven't done. I wish I had thought of it, to be honest, now that you just said that. The question becomes like, again, you get into the issues about the protein, right? So our expectations about the proteins that we're currently using, right, they're relatively short-lived. So I don't know how long I'd have to do it again, it's got to be some plasticity and change, right? And there might be, again, certainly when we take islets and just throw reg-3 gamma on them, we can see a improvement in glucose-stimulated insulin secretion. But that's probably not, again, that's different than the plasticity that comes from vertical sleeve gastrectomy that also seems to be dependent on reg-3 gamma. So I don't know, and again, it'd be really interesting to do it in a reg-3 gamma knockout, right? If I just threw some into the periphery and just kept pumping it for a couple of days, would I now get super good islets in a dish? And I don't know the answer to that. It's a really good experiment, and I hope you don't review the paper and tell us we have to do that. Because
Don't be worried. You talked about the stability of the red 3 gamma protein. Is an enzyme breaking down like DPP4?
Again, it's way more complicated than that, and I am not a protein engineer. It comes in different forms, right? Again, what we know, right, is that the forms that we're using here are not good antimicrobial peptides, right? And again, that has to do with, again, the way they're broken down, right? And again, in ways that are actually necessary in the GI tract, right? So where the GI tract is going to be a particularly good place to break things into the antimicrobial component. So, I don't think there's a DPP4 dependent function. That'd be sort of an interesting question because, again, my view of DPP4 inhibitors, right, is that there's still a lot of unknown components of what it is that we're actually doing with those. And so it might be useful to try with either a DPP4 knockout or using the inhibitors to actually see whether we can improve the effects of Reg 3 gamma in a way that I wouldn't have predicted. Now, again, I suspect that protein engineers will tell you we know exactly where DPP4 is supposed to act. I don't know whether we've done that analysis for Reg 3 gamma.
Okay, Helena Heldon, Umeå, Sweden. So in your EXTL3 beta cell knockout mice, you lost the positive effect on glucose tolerance with Reg 3. Was that due to reduced insulin secretion?
Yeah, the experiment. We haven't done the experiments in the way that you'd want in order to answer that question. But that would be the supposition, right? Is that Reg 3 gamma improves insulin secretion, and that's at least a big part of why it is that it improves glucose. And again, because we knocked it out there, that makes the most sense. But I don't have data on the levels to be able to show you that.
Can you just speculate on the translational component of this? Do you see this as a therapeutic?
Yeah. Again, based on the, I'll say this two ways, right? I don't know if Maury's still here, right? Or there he is, right? So I think what Maury would tell you, right, is what I showed you there is not a pharmacological effect that anybody would get excited about, right, from a therapeutic perspective. That is just not big enough, right, in the way we've done it. But that goes to the limitations of the protein that we're using and what we know about it, right? Can a better protein be better at it than what we have shown? But it also goes to the issue, like, how much of what's happening is happening in the lumen versus out in the periphery. And that's the part that we've been trying to get to as well, is trying to understand, again, the relative roles of circulating Reg 3 gamma versus stuff that's secreted into the lumen, because that would depend on where you want to target it, right? I certainly believe that the process of the plasticity, right, and its role in regulating the barrier, that there are therapeutic possibilities in that process, whether Reg 3 gamma turns out to be the thing that's targetable or not. But in there's lots that's happening that is important. And it's probably important, again, as a barrier, right? Better barrier, right? Less inflammation, less crappy stuff showing up at your liver. All of those things, right, are probably potential improvements, as well as, again, these effects on insulin secretion and other components of the system.
Yeah. Over here. Yeah, the other way. I'm Andreas, and I'm from the Camilla Scheele's group here in the building. And I was I can see it seems fascinating that just probiotics can increase this molecule, right? But what about in reverse? It's a little simplistic, but if you get antibiotic, do you actually see a decrease in Reg 3 gamma then? Because I guess that will also affect your response to gastric surgery if you were somehow on an antibiotic at the same time.
Yeah. So if Frederick Bachhead were here, he would comment on this, because we actually have sent surgeons at his group, right, to try to do some of these kinds of experiments. I can tell you that in germ-free animals, right, Reg 3 gamma levels are very low. And again, it makes some sense, right, because that the system is that Reg 3 gamma as an antimicrobial peptide, right? If there's no microbes, you just don't need very much of it, right? And again, as you know, those animals are weird, right? They're weird in their GI function in a variety of ways. And I think a component of that has to be associated with the fact that you're not provoking any of the regs and the antimicrobial systems. Again, it's not just antimicrobial, but important to organize GI tract function. So the issue about whether surgery depends on the biome, controversial. Again, I think as everybody here knows, right, again, who's played around with the biome, right, the experiments we do are somewhat simplistic compared to the complexity of the biome, right? Using antibiotics, germ-free animals, or even worse, right, using fecal transplants, right? I mean, that's a really gross, not just in a disgusting way, right? But gross in a scientific way to try to move. Because again, you're taking stuff out of the bottom of the tube, putting it in the top of the tube, and hoping that it all finds its way to the right location. So the point is, there's still a lot of controversy about this. So I don't want to sell short. I tend to believe, right, that there's absolutely a clear role for the biome to matter, but you will get pushback from different people around that.
Yeah. Hi. Up here. Thanks. Benjamin Jensen, UCPH. I was thinking about these antimicrobial peptides, we tend to call them host defense peptides now due to the wide sort of mode of actions that they have on the host rather than only on the microbes. But so what we often see when we work on different aspects of these many different host defense peptides available in humans and in rodents is that the luminal bacteria doesn't seem to change much, supporting that they have host-derived effects rather than microbial regulating. But when we look at bacterial translocation or encroachment, there seems to be a sort of very consistent effect across many different host defense peptides. So have you looked at these aspects, whether the microbes are closer to the epithelium or longer from the epithelium, depending on what model you use?
Yeah, so I am no microbiologist, so we have not done those kinds of experiments. It is clear that other people have done is that in Reg3 gamma knockouts, right, you get more infestation of the bacteria closer in than you would otherwise. But again, it's one of, as you said, one of many host defense components, right? So taking out one doesn't make the whole system collapse. But you're right. The issue about the segregation is really important, and the fact is, right, that these things matter to that level, right, again, of what's happening. But again, the notion that these are actually organizing other components of GI function, right, not just about the bacteria. That we found them because we spritzed them on bacteria and we killed them, right? But that doesn't define what their actual function in vivo really looks like. And I think you're highlighting that.
Right. We have a last question up here.
Just behind. Rag-free gamma is expressed by both intestinal and pancreatic cells. Which of the two contributes more to the phenotype PATQ1 observed in diabetes? Have you done the conditional knockout experiments on that?
Yeah. So we're building the flocks right now, right, so that we can do this. It doesn't exist. So it's going to take us another 18 months, 2 years to get to that. But it's the right experiment, right? And the question becomes, do you want to do it as a stop flocks, right, where I just ask about sufficiency when I turn it back on in different ones, or do I want to do loss of function, right, and take it out of different tissues? But again, it's expressed in lots of different tissues under different circumstances, not just the pancreas and the intestine too, right? So again, there's reason to believe that it matters for cellular stress responses in other tissues as well. So again, I think it's an open question. Again, we tend to focus on the GI tract because we see these nice regulations. But I can tell you, after vertical sleeve gastrectomy, you see upregulations of Reg3 gamma in the pancreas as well. Right? So it isn't just the intestine.
Randy, while I'm picking up the gift, we'll have one last question by Dr. Birnbaum.
Thank you very much, Maury Birnbaum, Pfizer. Great, a lot of really interesting stuff, and raises a lot of questions, of course, more questions. But one of them you now bring up is this interesting parallel between the effects of Reg3 gamma on intestinal permeability and its requirement for the efficacious effects of bariatric surgery. So there are ways of modulating, certainly genetically, gut permeability in mice. In general, how much of a role does gut permeability have in the positive effects of bariatric surgery?
So we have not tested, again, we've not gone to these other models, right, and start asking this question, but it's a totally logical version. I guess what I would say is, we know that hyperglycemia, right, results in poor barrier function, right? Again, it's part of what happens in diabetics who are uncontrolled, right? It's clear. So I don't think that diabetes starts with a bad barrier, right? I mean, I think diabetes starts with a bad beta cell, right? But that the negative feedback loop is fed by a poor barrier function, right, that contributes, right? So again, you have a bad beta cell, you get some bad glucose regulation that makes your intestine worse, that now contributes, right, to the downhill slide that this is. And that's the hypothesis we're working at, and that part of what bariatric surgery does is interrupt, right, that relationship, right? It's improving things at the beta cell, right, but it's also improving things at the gut. Right? And that's part of why it's more effective than most things that we do, because it's pleiotropic and doing lots of things to the system all at the same time. Great.
Thank you, Randy. And please join me in thanking Randy again.